might very well be the phospholipid. It's the thing that makes cells, right? So you can't have cells if you don't have phospholipids. The cell membrane itself is made out of phospholipids. All of the organelles inside are made out of phospholipids. The nuclear envelope is made out of phospholipids and endoplasmic reticulum. So they're very important. They are a absolutely essential portion of living things. But the problem is, on their own, they don't really do anything except for separate environments. So if you have a phospholipid bilayer and it's formed a nice little sphere, there's going to be a separate inside and outside environment, but they're not going to do anything. We know that living things can do stuff. They can reproduce, they can metabolize molecules, they can create enzymes that would help for reaction. But the phospholipid itself can't do that. It can't do any of those things. All it does is separate places, okay? So what we really need are these other things in the cell that will actually do things. And the things that do things in the cell are the proteins, okay? So today we're going to talk about proteins and how a protein is able to do things in the cell. First thing to know is, what is a protein? Oh, that's the problem. There we go. What is a protein? Proteins they're polymers, right? They are macromolecules, and they are made specifically out of amino acids, okay? So we can say that they are polymers made from amino acid monomers. Okay, and the important thing to realize about proteins is that proteins are going to have a shape that allows them to do their function. And their shape is always going to be a three-dimensional shape. Okay? So, for instance, if you need to do a task, you need to pick the right tool for the job. Okay? So, if I wanted to cut down a tree, I'm going to want to get an axe or a chainsaw or something like that. And it would be silly for me to go out with my water bottle and try to chop down a tree because it's the wrong shape. It doesn't do the right thing. Okay? So, proteins are going to have specific shapes to perform the action that they are trying to complete. Sometimes it's as simple as having a little pocket in the protein that fits in a certain molecule. Sometimes it's very complex. So one of the most important proteins, one of the ones that we're going to talk about the most in this class, is ATP synthase. ATP synthase has multiple different parts to it. One of the parts is a channel protein that allows hydrogen ions to pass through. But then the other part is like a turbine that physically spins and aids in the process of ATP synthesis, or making ATP. Okay? So they're like little machines that are inside the cell. Okay? So the three-dimensional structure of each protein allows it to complete a unique task. Okay? It's for this reason that a protein has to have a three-dimensional structure in order to be called a protein. We don't call it a protein until it's folded into its final shape. If it's just a string of amino acids, that's still a polymer made from amino acid monomers, but if it hasn't formed its shape yet, we just call it a polypeptide. Okay? So polypeptides are chains of amino acids that do not have a finished three-dimensional structure. Okay? So they're made out of these amino acid things. There are only 20 amino acids. 20 unique amino acids, which seems pretty amazing because we have all of these living things and they all have different proteins. and they do all kinds of crazy things, and they're all made from just 20 amino acids. But then that kind of is not that cool, because if you think about it, we've got all these different words in the in all of our languages, rather, and they're made from just 26 letters in the alphabet, right? So it's not out of the ordinary to say that there are 20 unique amino acids and they form all the proteins, especially since when you consider that most words are like less than 10 letters long, and there aren't any proteins that are 10 amino acids long. They're all significantly longer than that, okay? So let's talk about the amino acids and what makes an amino acid have a different structure, okay? So we can say that all amino acids share a common basic structure. And the common basic structure of an amino acid looks like this. The first thing you need to have is a carbon. You need to have a carbon in the middle, of course, because carbon is this awesome, versatile biological molecule and allows us to have a bunch of things bond to it. Okay, then we're going to look at the functional groups. The first functional group is on this side. Okay, and it's going to be an amine group. Okay, it's going to have nitrogen and then some hydrogens on it, like that. Okay, so we can say this group over here, that, oops, the nitrogen is also included in that. So this group here, we can call an amine group. It's a functional group, okay? Then, on this side, we're going to have another functional group that looks like this. Okay, and this functional group has a name. This functional group is called a carboxylic acid. Say carboxylic acid. Okay, so if you're looking at something that has this general structure here, and you're trying to think of a name for it, a name that might pop out in your head is amine acid, or how about amino acid, because it's got an amine group and a carboxylic acid group. Make sense? That's how they get its name. The amino acid is named because it's got an amine group and a carboxylic acid group in it. Okay? All amino acids have this structure right here. You might notice that the carbon only has 3 things bonded to it. That's because there's one more part. The other part comes down like this, and it is called the R group. Okay? The R group, or the side chain, is the thing that is unique for each of the amino acids. Okay, this is the part of the amino acid that makes each of the 20 amino acids unique. The side chain can do all kinds of stuff. It can be a side chain that is ionic. It can be a side chain that can form covalent bonds. It can be a side chain that is hydrophobic. It can be a side chain that's hydrophilic, right? So based on what this R group is, it's going to change the way that the amino acid acts as a whole. And then if you put that into part of a long chain, that's going to change the way that that chain acts. Okay? So we know that a polypeptide is a chain of amino acids, so we have to have a place for these amino acids to bond together. They're going to bond together using a dehydration reaction. Does anybody know what 2 groups are required for a dehydration reaction to happen?
O and OH.
O and OH. No, H and OH.
Oh, yeah.
Yeah, H and OH. So you got your OH group over here, right? And you got your H group up here. This is one bonding site. And this is another bonding site. So if you wanted two amino acids to bond together, it would be as simple as having the amine group of one bonded to the carboxylic acid group of another. And they'd form a nice long chain. Okay? And in that nice long chain, the R groups would all stick out from the side. Right? Yeah.
They flip over when they
You got these are all single bonds, so they're going to be able to spin around. Right? So, this bond, if this thing bonds with this thing, they call it a special type of bond when it's an amino acid. It's called a peptide bond. And if it's a peptide bond, that makes sense because we call these things polypeptides. And that means that there's many peptide bonds, or many amino acids bonded together. Make sense so far? Okay, so some little things about this. Interestingly enough, when you guys eat protein, then we're probably not eating proteins because proteins have to have their final structure. We're probably eating polypeptides because we've cooked them, and anytime you cook them, it's going to mess up their overall structure because it gets denatured in heat. And so you're going to eat a polypeptide, which is a long chain of amino acids, rather than eating a protein, which is in its final shape still. Okay. Other interesting things about this. Let's say I eat like an egg or something like that. Do I need egg proteins? Do I need protein? Maybe, right? What I need are amino acids, right? I need the things that the proteins are made out of. There's nothing in my body that's going to require a specific egg protein or a specific protein from a cow or a specific protein from a chicken. What I need are the amino acids or the little building blocks that will allow me to make my own proteins, okay? Now, what if I eat only one type of food and it's deficient in one type of amino acid, right? Am I going to be able to make my own proteins? Maybe not. Okay, so there are certain proteins, or certain amino acids rather, that can be synthesized from other amino acids. They can be converted, right? And so if you have a deficiency in one type of amino acid, your body can just take another type of amino acid and convert it into the right type, okay? But there are ones in your bodies that are called essential amino acids, and these ones that are called essential amino acids are ones that don't have the ability to be converted to from another type. Okay, so a lot of bodybuilders have to take amino acid supplements, right? Because they have amino acids, essential amino acids that are required for building muscle, that they'd never be able to eat enough food in order to get that amount of amino acid from it. So they just take pills that contain those amino acids so that they can have a supplement of that. Okay? Let's talk about the shape of a protein. Okay, there are 4 levels of protein structure. Okay, the first one is called the primary structure. Okay, and the primary structure is very simple. The primary structure just refers to the order of the amino acids. Does it go lysine, glutamic acid, valine, or is it glutamic acid, valine, lysine? Right? And so the changing of that very first primary structure is going to have a very large role in determining the other structures in this amino acid. Okay, so we can say the sequence of amino acids in the polypeptide. Okay, and this is obvious. If you relate it to language, we know, for instance, that if you wrote the word D-O-G, right, that's dog, and then if you move some stuff around, you can very easily replace that and be G-O-D or God, and those become very different things just based on the way that you order the letters, okay? So the way that you order the amino acids has a similar effect in that it's going to change the overall effect of the protein or the overall way that the machinery acts. Okay, let's look at the secondary structure. Secondary structure refers to hydrogen bonding that takes place between adjacent amino acids. Hydrogen bonding between adjacent amino acids. And it can lead to one of two different structures. Will lead to one of two structures. Okay, so the first thing that we can look at is where should hydrogen bonding be occurring? And we go up here. Hydrogen bonding occurs anytime a hydrogen bonds to a nitrogen, an oxygen, or those are the only things that are in there, right? So where is the hydrogen bonding going to happen? Yeah.
In the carboxylic acid on the oxygen.
Okay, but what happened to this when you
When
you have a bunch of them joined together, right? We used up this group and this group for the dehydration reaction, and that only leaves this one spot right here. So this is your hydrogen bonding spot. So this one spot is going to hydrogen bond with other adjacent amino acids, and based on which R groups will then cause certain structures. The two structures that can form are one that looks like this. This one is called the alpha helix. And you might see alpha abbreviated like that, a little fish. Okay. Well, the other one that can form looks more like this, where there's no overlap. It's just like a zigzag. And this one is called the beta pleated sheet. Okay, and the beta pleated sheet, beta might be abbreviated like that. It's a bee with a big tail on it. Okay, so they're going to form either the alpha helix or the beta pleated sheet. So we're starting to get a three-dimensional structure, and this three-dimensional structure is just going to be formed based on the hydrogen bonding of adjacent amino acids, which amino acids they are and what order they go in. Okay, so the primary structure is going to influence the secondary structure first of all, and then the secondary structure will go on to influence the next structure, which is the tertiary structure. And the tertiary structure is the most important structure here. The tertiary structure is the three-dimensional final folding of the polypeptide. Three-dimensional final folding that allows polypeptide to perform its task. Okay, and there are three, and so you need to put this in here, that tertiary folding is the result of R group interactions. Sometimes it's R groups interacting with each other. Sometimes it's R groups interacting with the environment. So here are some of the most common types of R group interactions. First one, something that we call hydrophilic interactions. Okay, so proteins are going to form inside the cell. They can either form in the cytoplasm, which is like the area that's in between the nucleus and the outer portion of the inner portion of the cell membrane. Okay, so that means it's not inside any organelle. Or proteins can be made inside of the endoplasmic reticulum. which is basically just another sack of phospholipid. But all of them are pretty much going to be aqueous environments. They're going to have water all around them. So if you have a whole chain of amino acids in a polypeptide that are all hydrophobic and then a long chain that are hydrophilic, it's very easy to see what's going to happen. If it's in a watery environment and there's a bunch of hydrophobics and then a bunch of hydrophilics, the hydrophobics are all going to clump together because they want to be around each other. And then they're not going to want to be around the water, so they're going to go to the inside of the protein, and the hydrophilic ones are going to wrap around and be on the outside of the protein. Right? So that's going to allow the protein to fold into some three-dimensional structure. Okay? That's the main driving force. It's also very interesting because if you take a protein and you denature it, which means that you mess up its folding, you get it back out into its long polypeptide shape, and you fold the exact same protein in like oil, like let's say we drop it into an oil environment, it will fold a completely different way because hydrophobic-hydrophilic environments or hydrophobic-hydrophilic interactions are going to do the opposite thing. All the hydrophobic stuff, when it's in oil, is going to want to be on the outside, and all the hydrophilic stuff will be on the inside, right? So based on the environment in which a protein folds, it's going to fold in a completely different way. There are some proteins that have to be folded using a little helper protein, and that helper protein is going to change the way that one protein folds. You know, it's going to say like, oh, I'm going to make this portion of the protein partially hydrophobic, so the hydrophobic stuff scoots over here. Okay? And if it doesn't have a helper, it's going to fold in a completely different way and have a completely different function in that case. Okay? You've also got ionic interactions. So if you had a R group that had an ionic negative charge, it's going to be attracted to an R group that has an ionic positive charge. Not only that, but it's going to be able to repel any other negatively charged ionic R group. Okay? Even if it's just a partial negative, even if it's just a hydrogen bond or something like that, it's going to be able to repel those partially negative charged groups. Right? The last thing is one of the most important things, and that is covalent bonding. There is a specific amino acid called cysteine, and cysteine is there in order to provide a sulfur group. Okay, so cysteine's got a sulfur group in its R group, in its side chain, and when two of these side chains get together, the sulfurs will bond together, and they'll form a covalent bond that's a very strong covalent bond, sulfur to sulfur, that we call a disulfide, because it's got two sulfurs, disulfide bridge. So it's called disulfide bridge. And disulfide bridges, their main purpose is to hold the three-dimensional structure together. So disulfide bridges hold the 3D structure together. Okay? So for instance, if I make some cool thing, I make a robot of some kind, and I just put the parts together, and I'm holding them, I'm like, okay, I got my robot here, everything's in the right spot, and then I let go of it, and there's nothing holding them together, all the parts will just fall apart, right? So the disulfide bridges sort of act like the screws or the nuts and bolts of these proteins that keep them in one specific folding orientation, okay? Lots of things can denature proteins. Heat denatures proteins. Acids denature proteins. So there was a question on your first reading guide that said, what's the significance of pH when we talk about biological systems? One of the big significant things of pH is that proteins will not fold correctly if they're not in the correct pH. So if they don't fold correctly, that means that they aren't doing the right thing. And sometimes, not only are they not doing the right thing or not performing their task, sometimes they can do very bad things. So I talked about this the very first day. Maybe it was just at open house. I don't remember. But biology is really cool. It's like the best thing to study right now because it's the thing that we're learning the most about. We, as a species, know very little about biology. I mean, you've got a textbook that you can read through, and that's like just skimming the surface of what we know. But What we could know is so much more than that. In fact, just yesterday, Scientific American published a review of a journal article that was published that said that we are now much more aware of something that are called prions, right? We were learning more about prions. And prions are misfolded proteins, okay? They're normal proteins that your body should produce, but they fold the wrong way. Okay, so normally we think, okay, it folded the wrong way, it's just not going to be able to do its job. These are specific types of proteins that not only do they not do their job, but they meet up with healthy proteins of the same type, and they cause them to misfold in the same way that they are misfolded. Okay, and so they can go around and they can make armies of these misfolded proteins that will then stop other proteins from working. We've known about prions for quite a long time. I think Nobel Prize was won for a paper on prions in like 1997, right? So that doesn't seem like a long time. I mean, it's like less than 20 years. But we're learning even more about them now. So the most famous type of prion is bovine spongiform encephalitis. That's what we call mad cow disease. And mad cow disease is transmittable only when cows eat the brain matter of other cows. Right? So you'd be like, well, that's never going to happen. Cows don't eat other cows. Cows are herbivores. Well, the thing about that is in the cow farming industry, the beef industry, the regulations used to be a little bit lax. And so what happened is in the slaughterhouses, they would kill the cow and they would cut off all the parts that like wouldn't be valuable. Okay, that was like ligaments and the brain. Nobody really buys cow brain. Nobody eats that, right? So they'd take all these parts and they'd throw them into a big grinder, right? And they'd grind them all up because it's made out of monomers, right? And the monomers could be used by the other cows, to make polymers of their own, okay? So they just take all this stuff and they incorporate it into the feed of the other cows. Okay, well, the problem was they were incorporating bovine spongiform brain into the feed of all the rest of the cattle. And so what happened is there was this massive epidemic of bovine spongiform encephalitis, which is mad cow disease, which killed large groups of cattle. And not only that, but in order to stop the spread, they just killed a bunch of cows and never used any of their meat because they couldn't be sure who had bovine spongiform encephalitis, and they didn't want any crazy thing to happen, like it gets transmitted to humans, right? So they just slaughtered all these cows and burned them, right? So that it wouldn't get transmitted. And that was a huge waste. It was a huge problem. The price of beef shot up. We wasted all kinds of resources and stuff like that. But we're learning about more and more prions. And we've thought for the longest period of time that the only people who were ever at risk for getting prion diseases were people who engaged in ritualistic cannibalism. Well, yes, it's true. If you are a ritualistic cannibal, then you are at a much higher risk. But we're learning now that certain neurological diseases are actually caused by prions. These aren't prions that you would get by eating other people. I mean, you could, but they're prions that are made because of something that happens in the body that causes a misfolded protein. And the misfolded proteins then can go on and misfold other proteins and cause these armies of misfolded proteins. There are a number of Parkinson's-related diseases, not Parkinson's itself, but a number of Parkinson's-related diseases that we have directly linked to certain prions now, okay? That means that under very strange instances, these are transmittable diseases, right? Some concern was brought up that surgeons are at a higher risk for getting these transmittable diseases because one of the treatments for Parkinson's involves implanting electrodes into the brain. And a surgeon has to do that. And so if the person is misdiagnosed with Parkinson's and the surgeon is then exposed to these prions because they're doing, brain surgery essentially, the prions are non-living things, right? So a normal surgeon is like, okay, I was exposed to bacteria, so I'm going to, go through this sterilization process. I'm going to use soap on my hands and things like that. But these prions are not sterilized by any sterilization process that we're used to using, right? That means that it's possible for it to end up on the surgical equipment. It's possible for it to end up on the surgeon's hands, right? So again, we're learning all this new stuff that we never knew of before, and we've got to modify what we do in order to kind of cope with what we're learning. But the problem with biology is we think we know so much, and every day stuff comes out that just tells us how much we don't know. Okay, we've never linked Parkinson's itself to any prion of any kind, but we've linked other things that look like Parkinson's to prions. Alzheimer's is another candidate where we think it might have something to do with a prion. Okay, it's not a prion that's transmittable that we know of, but it's a prion that's caused by some genetic marker, some genetic predisposition to have misshapen, misfolded proteins. Anyways, lots of very cool stuff that's happening right now. Where when I teach this class again in 5 years, I might have a totally different lecture to give now because we know so much more stuff. Okay. The last thing, let's talk about the quaternary structure. Quaternary structure of proteins refers to multiple different folded polypeptides that come together to form a larger protein. Multiple folded polypeptides that join together to form a large protein. Not all proteins have this. Not all proteins have a quaternary structure, but a lot do. We talked about one already in here that does, and that is hemoglobin. Hemoglobin is made of 4 separately produced polypeptides, 2 that are called alpha chains and 2 that are called beta chains, and they fold on their own, and then they come together to form one large protein, which is hemoglobin. Questions on this? Yeah.
So is it not like a real, like it's not a protein until it's in its tertiary structure?
It's not a protein until it's in its tertiary structure, and then if it's a protein that has a quaternary structure, it's not a protein until it's in its quaternary structure. So I think the term that I meant to use up here was that it's finished three-dimensional structure. So if it's a protein that that only has a tertiary structure, then its finished structure is tertiary. If it's a protein that has quaternary structure, its finished structure is in its quaternary. Yeah.
Going back to that, the amino acid picture.
Uh-huh.
For the remaining group, could hydrogen bonding be placed?
In the R group?
Yeah.
Yes. Hydrogen bonding in the R group happens a lot. Yep. Anything else?